So in continuation of our previous topic, validation of sterility. How to validate sterility? Yes, the confirmation of sterility. We have done definition, we have formulation, we have done sterilization. Now confirmation of sterility. So it has to be validated how it was done. Sterility testing for parenterals, because without sterility, the products are not marketed. They are not fit for in human consumption. That's why they go for microbiological testing. They take that sample, and then it has to be uh, applied on media. If there's a growth, it means there is a organism. If there is no growth, it means it is free from microorganism. So for that, testing attempts to detect the presence or absence of viable microorganism in a sample number of containers taken from batch of product. So what they have, they have taken various samples of batches of product, then they go for testing. Sterility testing attempt to detect the presence or absence of viable microorganism in a sample number of containers taken from batch of. So they have to take some samples of that, then test for sterility, the confirmation where there is a organism or there is a no organism. So presence of absence of viable organism in a sample, so that they must go for sterility to confirm that the product is sterile, free from organism, and it is a requirement of the parenterals. Overall, certain pharmaceutical products must be sterile. What they do? The drugs which are to be sterile: injections, ophthalmic preparations, irrigation solutions, hemodialysis solutions. Still, some more are required for this, so which are injections, ophthalmic preparations, irrigation solutions, hemodialysis solutions, whatever. These are must be sterile. Now, the two sterile products is those that can be sterilized in final containers, terminally sterilized, and those that cannot be terminally sterilized and must be septically prepared. So first, they prepared aseptically conditions, and first they must have to go for sterilization. In the final stage, they have to go for sterilized, and some are prepared under a aseptic condition, the environment free from microorganisms. So then they go for, what is the definition of sterile? Sterile must be free from microorganisms. Free from microorganisms. Free microorganisms. In practice, no such absolute statement regarding absence of microorganisms can be proven, defined as a probability of 1 in a million of container being contaminated. This is referred to as the sterility assurance level test. Sterility assurance level test. A sterilization process must deliver a sterility assurance level of 1 in a million. It is not possible to measure 10 to the power minus 6. The required SCL can be achieved by applying a process that will reduce the number of organisms to 0 and then apply a safety factor that will deliver an extra 6 log reduction. To the calculation they have to done. Validation, a biologic indicator, is a characterized preparation of specific microorganisms resistant to a particular sterilization process. So these has to apply these indicators, biological indicator. If they can grow over there, it means there are organisms are there. If no growth, means there are no organisms. A biological indicator, spores are added to a carrier, such as a strip of filter paper, packed to maintain physical integrity while allowing the sterilization effect. So they put some strips of growth media or papers. So if there is a growth, that can be found over there. Indicator, the spores are added to the representative units of the products being sterilized with assessment of sterilization based on these samples. So self-contained biological indicator, these are the different type of indicators which are used during the sterility test. Relating to the heat sterilization used in fermentation industries, the thermal death time they have to check, F value they have to check, decimal reduction time they have to check. Is the shortest time required to kill all microorganisms in a sample at a specific temperature and under defined conditions. It's a thermal death time is the time in minutes at a specific temperature, usually 250 degrees or 121 Celsius, necessary to kill a population of cells or spores. Decimal reduction time is the time required to kill 90% of the organism in a sample at a specific temperature. Selection of sterilization process must be appropriate for products. One is terminal sterilization is the method of choice. Moist heat is the most common process used for terminal sterilization. Product must not be affected by heat. Containers, closure, integrity must be established. Items being sterilized must contain water if sealed. Our material must allow for removal of air and penetration of steam for steam sterilizations. Protocol. Requirement for moist heat sterilization. One, validation protocol should include the following detail for each sterilization process. Process objective in terms of product type, container, closure system, SL required. Specification for time, temperature, pressure, and loading pattern. Description of all equipment and support system in terms of type, model, capacity, and operating range. Performance characteristics of all equipment, pressure gauge, valves, alarm systems, timers, steam flow rates, pressures, cooling water flow rates, cycle, controller functions, door closure, gasketing, and air brake system and filters. Methodology for monitoring performance of equipment and the process and laboratory testing methodology. Personnel responsible for all stages and final evaluation should have experience and necessary training and be authorized. Factor of importance in sterility testing is that the environment in which the test is conducted, the quality of the culture, conditions provided, the test method, the sample size, the sampling procedure. Sterility testing factors affecting growth of bacteria. There may be nutrition, moistures, air, temperature, pH, light, osmotic pressure, growth inhibitors. These are factors which affects the growth of bacteria. Sterility testing, what do we have to do? Pyrogen test, clarity test, leak test. This has to be performed. One, pyrogen test, clarity test, leak test. Immerse this ampoule in a dye, methylene blue. Apply vacuum inside. The ampoule of dye goes inside, means there is a leakage. Pyrogen and pyrogen testing produced mostly by gram-negative bacteria. So you can understand that pyrogen are mostly produced by gram-negative bacteria. Endotoxins, complex of pyrogenic. Lipopolysaccharides, a protein and inert lipids. Lipid part of the lipopolysaccharides is the main pyrogenic agent. So polysaccharides, water-soluble, thermostable, water-soluble, unaffected by the common bactericides, non-volatile. These are the reasons why pyrogens are difficult to destroy once produced in a product. So pyrogen and pyrogen testing, what they do? Source of pyrogen contamination may be from solvent, from raw material, from environment, containers and closures, personals, storage between preparation and sterilization can be possibility of the pyrogen. Test for pyrogens, first is the rabbit test, second is the LAL, Limulus amoebocyte lysate, Limulus amoebocyte lysate. This is LAL test. Two tests have to be performed. One is rabbit test, and they go for LAL test. Antipyrogen rabbit test. The test consists of measuring the rise in body temperature in healthy rabbits by intravenous injection of a sterile solution of the substance under the test. Why the rabbit? Reproducible pyrogenic response, similar pyrogenic response to humans, other species not predictable. So that's why they use rabbit for pyrogen test. Preliminary test, sham test, intravenous injection of sterile pyrogen-free saline solution. to exclude any animal showing an unusual response to the trauma, shock of injection, any animal showing a temperature variation greater than 0.6 Celsius is not used in the main test. Test of origin rabbit test is main test, group of 3 rabbits, preparation and injection of the product, warming the product up to 30 degrees Celsius, dissolving or dilution, duration of injection not more than 10 minutes, the injector volume 10 ml per kg of body mass, determination of the initial and maximum temperature. All rabbits should have initial temperature from 38 to 39 degree, 39.8. The difference in initial temperature should not differ from one another by more than 1 degree Celsius. The result of the Pargin test. If no rabbit shows an individual rise in temperature of 0.5 Celsius or more, the product meets the requirement for the absence of pyrogen. If any rabbit shows an individual temperature rise of 0.5 degrees Celsius or more, continue tests using 5 other rabbits. If not more than 3 of the 8 rabbits show individual rise in temperature of 0.5 or more, and the sum of the 8 individuals' maximum temperature rises does not exceed 3.3, the material examination meets the requirement for absence of pyrogen. So you can see that how they test, how much, if a single rabbit, how much rise, and then for 3, how much rise, and then they go for 8. So then they have to calculate rise of temperature of rabbits. If more than that means, if less than that means there is no pyrogen. If above test not passes, the sample has said to be pyrogenic. Limulus, amebicide lysate LAL, bacterial endotoxin test, an extract from the blood cell of the horseshoe crab, Limulus polyphemus. Limulus polyphemus contains an enzyme and protein system that coagulates in the presence of low level of the lipopolysaccharides. So this has to be done through LAL test. Preparation, manufacturer's area. So you can see the stock room is there, compounding area is there, clean up area is there, aseptic area is there, sterilization, quarantine, packing, storage and transport. So this is a parenteral manufacturing. So next we'll do about the parenteral manufacturing.